that articles.
6 articles this year, his first year.
Yeah.
And do you have a grant yet?
I have several smaller planning grants and some Amazon grants.
It's two. He has his FIAP, which is FSU is one of the few universities that provide what they call a first-year assistant professor grant, which is basically your first summer salary, like a full summer salary,
yeah.
right? And we have a lot of opportunities to get like planning grants and disciplinary grants, etc. That's another thing you might want to check into. And these are huge amounts, like 14,000, 15,000. In other schools, it's like, oh, we have this grant, and it's like 2,000, 3,000. Not as competitive as FSU. Anyway, without much further ado,
thank you.
Thank you.
I think some of you actually I've seen a lot of faces of you last year in my pro-seminar talk in this research seminar. I think it's research colloquium last year, and I was primarily focusing on my postdoc work. And I think because this is my second year at FSU, so I got opportunity to kind of advance some of my previous research. So I think for those of you who have graduated, who will graduate in the very near future, when you want to transition into an independent position, I can also share with you some of the experiences in terms of collaborating with different people from different disciplines, and as well as collaborating with students. I think.
Did you have a 2-year postdoc?
I have a 2-year master's student as a research assistant.
No,
He's been really grateful.
When you were doing your postdoc,
It's a year and a half. Yeah. And also during this year, I also got some opportunity to get some seed funding from both FSU Council, it's called Council for Research and Creativity, as well as Institute for Successful Longevity. And those initial grants actually allows me to hire some students to work with me on several components of these projects. So that's been really helpful. And some of this work you probably have already heard of it, but I just want to give you some background in case some of you haven't really get to know a little bit about it, so that I can transition into what I'm currently been working on. And the primarily focusing on the latter part of this presentation. This talk is about ontology-enhanced analysis of clinical research eligibility criteria. And in this presentation, I'll first talk about the problem of clinical trials and the generalizability problem. And also, I'll be talking about 2 different components that I'm working on. One is to analyze the quantitative eligibility criteria in clinical trials to see how I can leverage the informatics methodologies to understand the population representativeness of clinical trials. And then I'll be applying this on 2 different types of trials. One is on type 2 diabetes, another one is on colorectal cancer treatment trials. And then I'll be talking about my recent research on how do we identify restrictive qualitative eligibility criteria in clinical trials, especially on cancer. I know some of you might not be familiar with these terminologies, but I'll be explaining them during the talk. So hopefully by the end of this presentation, you will have a brief idea about what has been going on recently. So essentially the problem here, giving you a very big picture about about problem of United Healthcare. This picture actually I found it pretty astonishing. It shows that the United States, in terms of health expenditure, it's the highest among those developed countries. But however, the life expectancy is actually not that good. If you compare the United States with other countries like Norway, Switzerland, France, the life expectancy is much shorter. So we're actually really wondering, why do we spend so much money on healthcare, but why don't we have a longer life expectancy, and why don't we have a better health outcome? And the Institute of Medicine, which has been changed to the National Academy of Medicine, reported to the government in 2012 about the issues of the whole healthcare, especially what has been wasted. And they estimated like roughly $750 billion were wasted per year on healthcare. And they consider how much money would that be expended otherwise. So they investigate the whole process of healthcare from the bench side, which means that in the labs, for biology labs, for those medical school labs, they investigate whether a certain treatment or biological substance can be effective to treat patients. From that side, all the way till the bedside, like you develop treatment, you develop drugs to treat patients and for improving health outcomes. And along this whole spectrum, a lot of evidence were wasted and the experience were not collected effectively. So there are so many opportunities for us to look at what are the problems of healthcare and how can we improve those problems, how can we solve those problems so that we can have better outcomes and reduce cost of healthcare. So in my particular research, I was very interested in clinical trials. And the clinical trials are primarily like just testing whether a certain drug or treatment or medication or medical device, how they work in people, how they can improve like people's health conditions and treat some conditions. And normally, clinical trials is very time-consuming and costly process. It normally takes over 10 years to finish a single clinical trial on a drug, and it may cost over $100 million. And some pharmaceutical companies, as far as I know, they couldn't even support the whole process of clinical trials before they can go broke. I heard a lot of stories like they develop a drug and they put it into the clinical trial phase, and they couldn't, survive within the, 2 years, phase 2, phase 3. So they had to go broke, file bankruptcy because it's so time-consuming, so costly. But if the trial is successful, the benefit is huge because it can be used for patients, treating patients, and for various conditions. And the clinical trial, the major concern of clinical trial is recruitment. And a lot of trials, they couldn't get finished because they couldn't find enough patients to recruit. So, and you can see from this figure that the trial recruitment is actually, it spans over a very long time, and it's from phase 1, they recruit like 10 to 50 people, and then they test the drug. And then if the phase 1 is successful, they can go forward to phase 2 to phase 3 to expand their patient population to test. And however, even if you test your drug within 100 people, how can you make sure your drug is effective for 1,000 people around the globe or is in United States? So the problem of clinical trial generalizability has been a major concern in the clinical research community. And the problem is, if you cannot test your clinical drugs in a trial phase for a representative population that can represent the national population, if you put your drug into market and those people in the general population, they've got prescribed this drug and they will have a lot of side effects and adverse drug reactions, and some of them may be really, really serious. So some drugs will have, actually had to be withdrawn from the market. So you can imagine how this would significant, how significant the financial loss would be for those companies that support those drug development. And the FDA, the Federal Drug and Food Administration, they have this system called Adverse Events Reporting System, which is basically used for collecting information that are reported by those patients who, who use the drug. And if they have any side effects, they can report to this system. And they did some simple analysis to see how many adverse drug reactions were occurred every year. And you can see from 2006 to now, the number of adverse drug reactions actually increased every year. And after 2013, there's roughly over 1 million reports every year. So it's such a big problem that we cannot ignore. And we want to make sure that the goal of our research is trying to make sure that when you design a clinical trial, how can you make sure you don't ignore some population subgroups that may benefit, that may use this drug in the future. So you kind of test all the side effects and the adverse drug reactions within the trial design, within the trial phase. So that's a problem that we're looking at here. And the previous researchers have already reported the problem of this generalizability issue for various different kinds of trials on different disease domains. For example, in dementia, the researchers reported that the patients enrolled in clinical trials of dementia are generally younger than the patients in the general population. So which means that if you're old, you may have diabetes, dementia, but you're not testing in trials. So how can you make sure my dementia drug can be effective to you, right?
Why is that though?
Because most likely the problem is that older adults have not only dementia, but may have some other conditions. And those conditions may confound your experiments. And those confounding factors may kind of reduce what we call the internal validity of a trial. So in order to make sure that your design of experiment can kind of exclude those external factors, they want to have some more cleaner patients, more cleaner patient cohort. And however, those patient cohort are generally younger and they have fewer medical conditions that co-occur with the condition you are testing. So that's one of the reasons. So actually we're keep investigating what is that real problem. How can we better design clinical trials to kind of recruit those patients that even if they have some other conditions, those conditions may not directly interact with the drug they're tested. So this is
an answer to that?
There's a
insurmountable challenge.
Yeah,
it's true. You can't really control that.
Yeah, it's control. It's hard to control, but I see a lot of research going on, in different areas, like in biology, in medicine, and also in like epidemiology, using different techniques to tackle this problem. Because clinical trial generalizability is a major issue in clinical research. And a lot of people are doing this kind of analysis. And as I mentioned, this problem does not only occur in dementia, but researchers also reported this problem in heart disease, in like some inflammatory bowel disease, which is kind of rare, and also on cancer. Cancer is a major problem. A lot of people die from cancer. Cancer is a major, major condition that causes morbidity and mortality in the United States. So, we cannot ignore this issue. We have to see how can we better design trials to make it more generalizable. But by the meantime, making sure that my internal validity of trials can be also ensured. And also, general public also reported that roughly 53% of new cancer diagnoses are in people 65 years or older, but those age group only accounts for 33% of the participant in cancer clinical trials. So in my research, we are really trying to use public data sets that can be used to analyze use public data sets to do some experimental analysis, do some empirical analysis to see what's the problem of trial design and how can we improve trial design. So we're using this data set called clinicaltrials.gov. This is a website it's basically a registry, a clinical trial registry in the United States. And all the FDA-regulated drug trial, device trial or biologic substance trial have to register into this website before they can recruit any patients, right? And even after, right now, the more regulation coming out, they're saying if you finish your trial, you have to report your basic characteristic to enroll patients into this registry within 24 months of completion. And and also they try to reinforce publication reinforcement because they want to say, okay, if you have finished a trial, we have spent so much money on you, you have to let the public knows what's going on in the trial. You have to let them know, if your trial is successful or not. But because publication is a very tricky kind of business. Like normally only positive results got published. The negative result normally they don't get published. So that's why the government regulates that, mandates that those trials, even if you fail, you cannot finish. You still have to report the basic characteristics of patients that you have enrolled into this registry to allow others to look at it and investigate it. So right now, I just looked at the statistics before I came to this presentation. There are right now over 230,000 clinical trial summaries that are conducted, of those trials are conducted all over the world in over 190 countries. But most of the United States trials have to be registered here. And this trial registry, actually, I've been talking to Lynette a lot about it. We're always saying, okay, this registry, even if it has a lot of information, but however, the information is really hard to read. And there was a reported article about the readability of this registry, because initially this registry is not only for the professional use, they're also trying to allow the consumers or health or patients to look at opportunity for clinical trial to enroll. Because many patients, if they cannot afford the treatment, if they don't have any health insurance, they cannot afford treatment. But if they can find a good clinical trials, they can enroll in the trial and get free treatment. They'll probably get treated, right? So they want to look for opportunities, but the readability is very poor. Previous research reported that in order to understand the trial descriptions in this registry, you normally need to have about 20 years of education background. So that's, not even PhD will not be enough. So we're really interested in how to make this registry more accessible to normal patients. So anyway, if you look at a specific trial summary, there are some information which are structured, and those information are really easy to read and also easy to extract. The information like the trial status, saying whether it's still recruiting patients or whether it's completed, is there. And also the condition, what kind of condition this trial is tackling is also here. The intervention that are being tested in trial is also here. And also some study design issue, like whether it's randomized or non-randomized, it's masking or non-masking, something like that. So, but that's not our focus here. Our focus is here in the clinical trial eligibility criteria. This section is a very important section in clinical trial summary because it characterizes what kind of patients can be recruited in clinical trials. And this section is largely unstructured, which means that most descriptions are written in a free text manner, in an open-ended manner. So you can use you can basically apply the same criterion, but you can say it in 1,000 different ways. So the challenge is actually trying to understand this text using computer methods, using natural language processing, so that we can do some analysis on the fly. And if we can actually broadly categorize this criteria into two categories. Like the criteria, like saying the hemoglobin A1C should be less than 10%, like body mass index should be greater than 35 kilogram per meter square, and the age between 18 to 60, and those can be considered as quantitative criteria, specifying a value range, and whether your specific value can fall into this range will determine your eligibility. And also there are some criteria that are qualitative, like you have diabetes or not, and whether you are using insulin, and whether using some medications, or do you have any comorbidities that may affect your life expectancy, or some other comorbidities that result in ineligibility for gastric bypass surgery. Those are somewhat qualitative. There's no quantifiable metrics to assess whether you can be eligible or not, but they're using pretty much binary decisions whether you meet this criteria or don't meet this criteria. And we need to tackle these different criteria in different ways because, remember the goal of the research is trying to understand those texts and be able to characterize whether the patients that are enrolled in trials can be represented to the general patient population. So we need to really trying to tackle this problem by, kind of break it down to sub-problems and tackle them.
I mean, you can certainly ask those questions in a closed-ended type of question. Do you use insulin regularly? I mean,
right.
Why is it open-ended to begin with?
I think this is a very good question, and a lot of researchers they always want to make it more structured, asking some open-ended questions. But however, some of the questions are really not easy to structure. The criteria I show you here is very simple question, very simple criteria. But in the later slides, you will see some really complicated situation. They may say, okay, we don't want to have patients with a heart attack within the last 6 years. But besides, a certain situation. So those kind of contextual information, those additional criteria that are somewhat related to the major criterion is very difficult to structure.
So not just answerable by yes or no.
Yeah, it's very, very difficult to, to let them fill in the blanks or, specify using a drop-down box. So, so that's poses a challenge for researchers to analyze those things in a systematic manner. So before we go ahead with our specific techniques to tackle this problem, I want to kind of give you a broad picture about what kind of population we're looking at here in this analysis. So the target population represents those patients to whom the results of your trial should be applied. It means that, for example, if you're designing a clinical trials on type 2 diabetes, you probably want to make sure that your target population are those type 2 diabetes patients because those people will be likely using this drug. And the study population represents the patients that are being sought after in the clinical trials. And they can be categorized using clinical trial eligibility criteria because it basically, specifying what is the population that those clinical trials are looking for. And however, because of some real challenge, especially as I mentioned before, a lot of trials they cannot finish because they cannot recruit enough patients. So the patients that actually can be recruited are recruiting clinical trials is only a subset of those study population, and they may not even represent the study population because if you are, for example, if you are FSU and you want to recruit patients, you could probably only get patients from Tallahassee area because if they are so far away, they cannot travel here, and also they may not be interested. So your population in this area may not be representative to the population. So the study sample is even smaller subsets. So In terms of assessing the generalizability of clinical trials, some researchers have used different type of metrics, and we can broadly categorize them into a posteriori generalizability assessment and a priori generalizability assessment. And the posteriori generalizability assessment is basically using the publication results of clinical trials, because in those publications, they may say, okay, what kind of population I have already recruited. Like, for example, if I have recruited 100 patients, they reported their age values, a mean age value, standard deviation, and specific variables. So you can use that to see, okay, what population have been recruited. And then you can use like national population survey, or you using your medical record data in hospitals to look at what population are like for those patients with a specific disease and do the comparison. And this can be only done after trial completion, because before the trial completion, you don't even know what patients have been recruited or not. And that kind of delays the process, because after the trial is completed, why do you need to, to look at what's the problem with your trial design? It's done already, right? And we want to improve the design, because before the trial is starting to recruit patients, we can try to see how can we improve the trial design to make it to make it more generalizable before they start recruiting. And the traditional methods, usually they look at the trial protocols, and they look at each criteria by each criterion by criterion, and they assess whether this exclusion criterion can exclude this many patients. So they can kind of, in an ad hoc manner, they can look at what population would have been excluded by this criterion.
Incorrectly excluded. Right?
Yeah, incorrectly excluded. But I'm not sure, what's a common practice of clinical trial design, if they really do this exercise to look at, if they apply this criteria, how many patients will I excluded. I guess this is hard, because, even for being able to identify patients that meet a certain criteria electronically is another challenge, in the medical science community, in the medical, because in the clinical, hospitals, it's very difficult for them to identify from the medical records what patients can be recruited or what patients can be potentially eligible. And that's kind of another research line called clinical phenotyping, using medical research data, medical record data to identify if a patient has a certain condition or not. That's another challenge. So in this research, we're really trying to use the clinical trials protocols using the eligibility criteria design to profile the study population. So we don't have to compare the study sample with the target population. We can make this process earlier in the trial design phase, so that when we design the study population with the target population, we can assess what population would have been underrepresented in this clinical trial population. And I mentioned before that one of the challenges is to unify those different representation of the same criteria into the same format. For example, the hemoglobin A1C, this is a very important metric for assessing whether you have type 2 diabetes or not. And they have different representations. One people can say HbA1c, another one can say hemoglobin A1c, another person can say glycohemoglobin, another one may say A1c. So, it could be possible for human beings to unify them, but for computers it's difficult. So we need to leverage existing dictionaries and using some manual coding to kind of tell the computer that, okay, these representations mean the same variable. So we need to use the same variable and use the same measurement units to unify them. And imagine in medical science, there will be a lot of different variables. Like, for different disease, there could be so many variables, and they may using different measurement units. So this task itself is also a very challenging task. The good thing is that we have, in my previous postdoc lab, we have a postdoc who developed a tool called ValX, which is to extracting those, which can be used to extract those numeric expressions from free text and unify them into the same name, into the same variables, name and also the measurement units. So we can do some simple analysis, like we can say for all the type 2 diabetes trials, how many trials are using 7% as a threshold value for this variable, and how many of them are using 10% as a threshold for this variable. And this can be done automatically and computationally. We also have developed a tool called Vita that actually accessible for everyone to be used to see what populations what populations are being recruited by clinical trials using this analysis. And this is a database structure that we use to structure the clinical trial summaries. Basically, recently update was September 2016, because every time we update, we need to do a whole lot of transformation, and it takes time. So I don't usually spend too much time on this. So I usually do this twice every year. And the updates of clinical trial summaries actually occur on a daily basis. So we try to keep track of the new trials when they come to the registry, but it's not going to be possible for us to do this very, very quickly and very frequently. How much time do I have? All right, so quickly reporting some of the results of this quantitative variable analysis. In order to assess whether type 2 diabetes trials can be are representative to the national population, we use the national survey called the National Nutrition Health Examination Survey. This is a survey conducted by CDC, and they publish the results. CDC is short for Center for Disease Prevention and Control, and they publish this survey on a 2-year basis. So every 2 years, they kind of surveyed roughly 15,000 people around the country, and they use very complicated sampling methods. And so after they sample these patients, they give them a sample weight, and the sample weight represents the number of patients for that each patient can represent in the national population. It's basically using US Census data to make sure that the patient data can be representative. So we identify those type of diabetes patients from these data sets, and we profile the target population. And from the clinical trial summaries, we identify all the interventional trials on type 2 diabetes to profile the study population. And this is how we identify patients from this survey. They didn't tell us whether it's type 1 diabetes or type 2 diabetes. So we need to use some methods to exclude the type 1 diabetes. Normally, type 1 diabetes patients got this disease in a very young age, very childhood, because they were kind of born with this disease. And normally, they are insulin dependent, which means that they have to make sure that their glucose value is low using insulin. And this one, so type 1 diabetes is usually called insulin-dependent diabetes. So excluding those patients, and we found roughly 2,000 samples, and if we use sample weight, we can extrapolate it into about 15 million patients in the country. So this figure shows the type 2 diabetes patients in terms of age value. What's the difference between the trial population and the real-world population? In this trial population, we found that most trials actually recruiting patients in a pretty broad range of ages. Normally, they don't pose a significant restrictions. You can see this threshold is 80%, which means that roughly 80% of trials recruit patients between 20 years old to 70 years old. But in the real world, the patients are generally older. Like the peak is 65, 66, and 80 years old. And but if we compare these results using HbA1c, which is a hemoglobin A1c value, we found there are some gaps. In the real world, the patient's HbA1c is is lower than the clinical trial population. So there is a significant gap between them, right? And if we look at BMI, these three curves actually aligns better. It means that with respect to this variable, the trials of type 2 diabetes are doing a pretty good job. So we previously developed a metrics to assess the difference between this trial population and the real-world population. And I won't go too much detail into this metric. Basically, given a set of trial, given a set of patients, we can use this real-world patient to assess how many patients would have been included in the trials, using that metrics to quantify the representativeness. So we can compare the difference between different sets of trials. And we use that to compare different phases. As I mentioned, usually phase 1 trials recruited very few patients, and they enlarge their patient population as the trial progresses. So the age variable, the G-score actually increases from phase 1 to phase 3. But however, the HbA1c value, the G-score decreases from phase 1 to phase 3, which kind of hint us that maybe when they enlarge clinical trials population, patient population, they might go to more healthier patients than a real-world population. So that's a very simple observation we can make. And recently, we applied these same methods on a completely different type of trial called the colorectal cancer treatment trial. And the reason for that is that I want to test this method in EHR data, medical record data, and I have a collaboration from University of Florida, and they have a teaching and research hospital called UF Health Shands Hospital. And in this hospital, they have a huge database called Integrated Data Repository. Right now, they have roughly 700,000 patients data in this data warehouse. So we identify all the colorectal cancer colorectal cancer patients, and using clinicaltrials.gov to find 1,300 colorectal cancer treatment trials. And we do the analysis. I want to see, in terms of G-score, how good are they or how bad are they. And this is our patient population from UF Health. And we found 3,000 patients with this disease, and they're roughly pretty divided between male and female, but in terms of race and ethnicity, most patients are white, and followed by Black and Asian. I guess this population.
Does that mean that they oversampled on white?
I guess it's not oversample. It basically means maybe more white people go to this hospital. So you know, that kind of can be used as a limitation of this research, because we probably cannot say that, you know, in terms of representativeness, we cannot say these colorectal cancer trials cannot represent the general population. We can only say how can it represent the population in the UF area, in Florida area, at most. So we look at 5 variables like age, creatinine, platelet count, ALT, and AST, and those variables are frequently applied quantitative variables in these cancer trials. And we calculate the G-score, and this is the distribution of these populations. And in terms of creatinine, this is a trial population, and this is a real-world population. They are somewhat pretty well aligned, and this is the age. And most trials are accepting age, also for a very wide range. And the real population, the cancer patients are generally like this bell curve. And we used G-score to do this calculation as well, and we found that G-score actually are doing pretty good job because their values are pretty high compared with type 2 diabetes. So we can roughly say that in terms of quantitative variables, those trials are doing pretty good job. They're not really restricting patients using quantitative variables. But however, they could use qualitative variables to restrict patients. So previous research did by Lewis et al. In a Journal of Clinical Oncology, they reported in cancer, they did analysis on over 600 cancer trials, and they found some restrictive qualitative criteria that may restrict the participation of older adults. So this is what they reported, and we try to identify the occurrences of those qualitative criteria in colorectal cancer trials. And when we did this research, we couldn't do this analysis on a very systematic and on all the trials. What we can do is that we can only look at the keywords for those like congestive heart failure, myocardial infarction, or heat stroke. And we found within this 1,300, 600 trials contain some keywords in this qualitative criteria. And when we selected some random sample from the 600 trials, we found that 44 trials had 76 occurrences of the keywords of this criteria. And so this analysis only is about 100, within 100 trials, we found so many restrictive criteria. So we are really wondering how these restrictive criteria are, if they are prevalent in all the cancer trials. So the answer to this question is that we have to develop some tools to parse those free text criteria so that we can analyze that.
They're not even mutually exclusive.
No, not really mutually exclusive. But they may be only used in one trial. For example, a trial may use 12 months, another trial may use 6 months. And of course, 12 months is more restrictive than 6 months. So we can even use the metric to kind of using some quantitative metrics to assess how restrictive certain qualitative criteria is. So I'm going to talk about how can we tackle the problem of parsing those qualitative criteria. It's also very difficult. There are a lot of challenges in the natural language because in order to understand the natural language, it takes a lot of efforts and and methods. So in terms of challenges, I only present here 2 major challenges. And the one challenge is that there could be using different terms to represent the same concept. Like they could use heart attack or myocardial infarction to present the same thing. And also they may use very complicated sentence structure. So here I list just 2 examples. And then one example is that it's no history of drug, alcohol, or substance abuse within 6 months before screening. So within this sentence, there are actually 3 major criteria: drug abuse, alcohol abuse, and substance abuse. So we need to decompose this complicated criteria into 3 major concepts. And also for this one, prior invasive malignancy, except non-melanoma skin cancer. And this is actually additional information. And also it has a temporal constraint saying unless disease-free for at least 3 years prior to registration. So we previously have used some techniques of language to extract concepts. We call it n-grams. N-grams is a contiguous word of n words. So n-grams, if we use 2 grams, it means 2 contiguous words. But you can see here, if we use n-grams, it doesn't work because it's not contiguous. We have to identify, okay, this drug is describing this abuse, and this alcohol is describing this abuse, and this substance is describing this abuse, to use that information to decompose. So we have to develop some new tools, and this is what we come up with. We are still in the process of developing this methods because it takes a lot of iterative modification. And what we did is we took all the clinical trial summaries on cancer, 4 different types of cancer: lung cancer, prostate cancer, breast cancer, and colorectal cancer. And then we performed some sentence splitting, tokenization, syntax parsing, dependency parsing. I'll show you what do they mean in a few slides. But essentially, we're using Stanford NLP toolkits to parse the sentence of criteria, and then we're developing some new components to recognize those major concepts as well as its contextual information using both pattern matching and the named entity recognition. So, and then after we do this whole analysis, we can analyze the temporal constraints that are applied on the criteria in a more systematic and large scale. So first of all, about this pattern recognition. So we can see from the sentence that a lot of these sentences about criteria are actually following a similar lexical pattern. For example, this sentence called any history of hemoptysis within the past 12 months, they have keywords like history of, and they have another keyword within, and then between this history of and within, there is an area in which there might be some major concepts, major criteria. And then after this within, it follows a duration. This duration is a temporal constraints that are applied on this criteria. So we can predefine some lexical patterns that can be used to identify those criteria with temporal constraints. Like we have actually identified roughly 17 temporal patterns, but actually there could be more. We're still in the process of enriching these lexical patterns so that we can better structure the criteria. So some criteria like pattern like within, the example sentence is like no myocardial infarction within the past 6 months, and more than patients must have been off previous anti-androgen therapy for more than 4 weeks, and etc. So we're trying to use the lexical patterns first, using regular expression to identify those criteria and then structure them. So this is what a lexical pattern looks like in this format called token regular expression. So history of and within, there are keywords, and then between this history of and within, we try to identify 12, we allow 20 words, in which in these 20 words, we're trying to identify concepts. And then after this within, it follows by duration. And those duration can also be recognized by the Stanford parser. And we annotate this area called the text NER area, named entity recognition, and we annotate this one with duration so that this sentence can be automatically recognized as different components. So it can do different things on different components.
Concepts don't necessarily need to be contiguous?
The concept doesn't necessarily need to be contiguous. I will show you next slides how can we tackle with non-contiguous concepts. So this is how we use the lexical patterns. So next is the name recognition. In order to identify concepts using medical domain, we use a dictionary called Unified Medical Language System. It usually can be considered as a lexicon that it can be used for recognizing concepts and the entities. And it's a very large dictionary. It contains over 3 million concepts and over 9 million terms. So normally, what kind of terms you are using normally are included in this data set, in this dictionary. So we can use that to detect whether a noun or noun phrases are concepts in the medical domain. So, and also they define some semantic types. So they assign those concepts to semantic types so we can restrict our concepts to a certain semantic types. Here in this research, we only are interested in the criteria about disorders and procedures and about clinical drug, because those are major criteria that are applied in cancer trials. So those are specific types, including like disease or syndrome, neoplastic process, silent symptom, et cetera, et cetera. So anyway, I want to spend a little bit time to talk about how we parse the complicated criteria. For example, this sentence that I just mentioned before, called no history of drug, alcohol, or substance abuse within the 6 months before screening, and we use the dependency parsing from Stanford to recognize the dependency between their words, and then using the dependency and using the, because also like this NLP tool help us recognize there is a conjunction within this area. So we can use this conjunction and identify the sub-area with noun phrases and decompose this area into 3 terms. and recognize medical concepts within these three terms using dictionary matching. And this area is also recognized by the Stanford NLP as a duration, so we can directly use the results of that parser. And also because we cannot only purely rely on the dependency parsing, we also use the syntax tree parsing. Syntax tree is basically trying to detect the syntax of each word and detecting the the noun phrases and their hierarchical structure. So, for example, for this sentence, like patients must not have a history of bleeding, diathesis, or have used anticoagulant therapy within 10 days of study entry. After with the Stanford parsing, they can come up with this syntax tree. And in this syntax tree, every word is tagged with whether it's noun, whether it's verb, whether it's adjective, or et cetera, and also they have dependence, they have a relationship between them. So we kind of traverse this tree to find the longest noun phrases, and then using this noun phrase to recognize whether this set of words can be matched to a term in a dictionary. And if this term cannot be matched to a dictionary, we go down the tree to find the subterms within these noun phrases. So, for example, for this particular noun phrase, we can identify it to be a humanized concept. And for the other noun phrases, if the humanized concept can be identified, we'll just identify it. If not, we'll go down the tree to find some specific substrings that can be recognized. So we actually recognize two different concepts from this sentence. So in terms of data sets, we use roughly 10,000 trial summaries on lung cancer, breast, prostate, and colorectal cancer. So most of them are clinical trials, interventional studies, and a few of them are observational studies. And some trials we look at also, for those trials, we'll also look at how many of them are how many exclusion criteria and inclusion criteria do they have. And also from inclusion criteria and inclusion criteria, they may also use, sometimes they use negation or non-negation, because they may say, okay, in the inclusion criteria, they may say we don't want patients with a heart attack, and the exclusion criteria, they may say history of heart attack, and they mean the same thing. So this is the frequency of the lexical pattern. So the most frequent pattern are within, followed by more than, within, prior to, etc. And this is the semantic types of the criteria. And most criteria about disease or syndrome, followed by therapeutic preventive procedure, followed by diagnostic procedure, and followed by laboratory procedure. And this is the kind of like very big results about the frequencies of the criteria and also the temporal constraints. The most used criteria is cancer. And this axis shows the temporal constraints of months that are applied on this cancer criteria. And most of them are 60 months, which means that most criteria say we don't want to have patients with cancer within the last 5 years. And just showing some simple results. Looking at this figure, we found that those criteria about conditions, disease, are often applied with relatively longer temporal constraints, like 6 months or 12 months. And the criteria about the procedures and the therapeutic procedures and the chemotherapy, they're always mostly applied with a very short temporal constraint, like 1 month. So here are some examples of cancer temporal constraints that are applied on cancer criteria. Like this one saying no other invasive cancer within the past 5 years except superficial melanomatous skin cancer. And this one saying previous cancer with terminal prognosis less than 5 years. And another one say history of another cancer within the preceding 5 years. So we can use our methods to extract the concept cancer and also its temporal constraints 5 years from these sentences. And we develop a tool. I hope I can show you a little bit about the process of running this tool. So essentially, in this front end, a user can choose a certain type of disease, and then we allow user to choose a certain type of clinical trials. Like they can specify that the trial should be conducted between 2000 January to November or December 2016. And then they can specify that the trial should be interventional, using drug as intervention. And then after that, the user can click process so that we can tell the user what are the frequently used qualitative criteria that are used in this type of clinical trials. And we can choose different numbers of features we want to show. And then it takes a little bit to, for the system to analyze the frequent features among those trials. And after that, a user will be able to select certain trials to do some further analysis. And this one shows the frequency of those criteria that were identified in this criteria sentences. So if you scroll down, you found this box, and you can just click on certain criteria like a chemotherapy. And if you click process, we'll show you the temporal constraints that are applied on these criteria in all the lung cancer trials, and also the temporal patterns and some co-occurrent features that are used along with this feature. So most of this, actually most of the trials do not apply any temporal constraints. They just say, okay, we don't want to patients with chemotherapy. And if they have some temporal constraints, some of them use 1 month, some of them use 3 months, etc. And as I mentioned before, most of them don't use any temporal constraints, so they have no patterns. And this one shows the other co-occurrent criteria that are used within this set of trials along with this criteria. So we can export this result into a CSV file, and then the user can just click on this like a CSV file to find the trial number, so that they can go to the clinicaltrials.gov to look at the specific trial, to look at more information about that. Okay. So also this is very, it's also publicly available, and Lynette actually helped me with doing some preliminary assessment of the usability, and we'll keep working together on this part. That's great. So yeah, this figure is for another feature, myocardial infarction in lung cancer. We found that most trials are using 6 months for this criteria, followed by 12 months. And I just quickly go through them. And also because this is computational work, we want to evaluate the methods, whether it's what's the performance like. So we can evaluate in the different aspects in terms of coverage of those lexical patterns, and in terms of name identity recognition, whether we actually extract those concepts correctly. And then also we can extract the temporal constraint and also usability of a front-end tool. So in terms of evaluation for the lexical patterns, in order to do that, we randomly selected 500 eligibility criteria from our data sets, and we categorized those qualitative criteria with temporal constraints that have been correctly matched to one of our predefined criteria pattern as true positive. And for those criteria that cannot be matched to any predefined criteria, we call it false positive. And if some qualitative criteria, if they don't have any temporal constraints, but they cannot be matched to any temporal patterns, it's false negative. And for those qualitative criteria without temporal constraints, or if there are quantitative criteria that were matched to a certain pattern, it's false positive. So we manually look at all these 500 criteria manually and compare it with our methods. And the recall is roughly 80%, and the precision is 94%. So the F measure is 86%. So we can see here we kind of like have a pretty good precision, but we're a little bit worse in recall, because there are like tens of thousands criteria, and it's really difficult to find extensive, comprehensive list of lexical patterns that can identify all these criteria. So I will show you some simple results that cannot be identified within our problem. So one problem is actually due to the problem of this Stanford NLP parser. For example, this sentence, history of myocardial infarction or unstable angina within the last 12 months prior to study enrollments. The NLP methods of Stanford identify this last 12 months as a date instead of duration. So because we have to identify duration in order to match it to our pattern, so it cannot be recognized. But if we delete this word last, the Stanford NLP can recognize it as duration. So it shows you that because we have a really flexible natural language, so some of them cannot be correctly recognized. And also there are some problems with free text criteria, because some of them are used in a really free manner. And for example, this one using patients with 24-hour urine protein greater than or equal to 500 milligram, or a history of primary renal disease. And in our pattern, we didn't use with the duration, because we usually say within or less than or greater than. So we're looking at how many trials are using this, following this structure, to see if we need to include this pattern or not into our criteria. And also another example of problems is that in this criteria, one cycle of intermittent therapy up to a maximum exposure to 6 months. This one is also not really commonly used, pattern in the this sentence is not very, commonly used in criteria. It's pretty ad hoc. So it's pretty difficult to identify all the criteria with temporal constraints using lexical pattern because they are rule-based methods. Rule-based method, you always suffer from recall, but giving preference to precision because precision can be ensured using those patterns. But however, because even if we cannot detect it to be a lexical pattern, to match our lexical pattern, we still can use a tool to extract concepts from it and also extract iteration. So it depends whether we want to look at those criteria that can match a pattern or not match a pattern to see if we want to use the results or not. So in terms of limitation, because we're kind of a part of this work is relies on the accuracy of standard parser. So the parser, they may not correctly identify a syntax of a word, or they may not correctly identify the dependency of a word, so there might be some problems. And also, because our lexical pattern, it's hard to make it so comprehensive, so we're still working on it, making sure that it can capture most of the trials that we want to, most of the criteria that we want to categorize, recognize. And we found that procedure criteria often applied with a short temporal constraints, and the disorder criteria often applied with a relatively longer constraints. And the effective use of causal health data repositories is promises to improve the transparency of the design issue of clinical studies. And finally, I would like to acknowledge some people working with me on this research, including my RA, Zhiwei Chen from computer science, Lynette, who is sitting here. He helped me with the design of the interface. And Ji Wang is a statistics PhD student working with me on another line of work, analyzing a large number of patient data. And Sarah Mixon is a Europe student, undergraduate student in biology, working with me in manually look at those criteria to see if they extract correctly. And also I acknowledge my collaborator, Dr. Zhang Bian from University of Florida, Henry Carreta, he's a geriatric research expert in FSU, and Tom George is a cancer researcher in UF, and Gloria Lipori is I think she's a database administrator in UF Health, Shanghai Hospital. And also previous research, including the tool of compact and the database compact and the tool VITA were developed under the support of online grants. And currently the Ultra and the VATEC tool are supported by 2 Amazon research grants and a planning grant of ISL and also my FIAP grant, and also part of it because my collaborator Ben Zhang, he's working at UF, so he's supported by this INH NCATS grants. So that's it for the presentation. Thank you. It's long.
We have a 2 o'clock class.
Yeah.
So we have 2 minutes if you have any questions.
Yeah. I'm sorry to put so much material into the same presentation. I always do this, so it may be a little overwhelming. Yeah, feel free to contact me if you have any questions, if you want to talk about the potential research projects, or if you're interested in using NLP to do some of the work, to try to automate and save some human experts or human efforts.
Using his expertise, collaborating with with
Sang-Hee, yeah.
On looking at social Q&A. So these methods can be applied to various types of qualitative data. So not just clinical trials.
Yeah, we're right now having 2 different projects going on. One project is really interesting. We're looking at the cancer Q&A data, and we're looking at using a panel of patients. Like for each specific patient, we look at his questions longitudinally to see what kind of questions a person has asked during a certain period of time to see if these questions are aligned with his progression of the disease. And I'm working with both Sang-Hee and also Professor Dr. Zhou Ho from Communication School, FSU, in this research. Thank you.
Thank you.
Thank you.